Welcome to Lab 5. In today's lab session, we're going to be discussing about two types of specialized connective tissues, cartilage and bone, and we'll also cover bone formation. This was covered in three lectures, so today's lab will be a longer one. So first, let's have some summary questions. Generally, in these summary questions, if you need more time, please feel free to pause the video. So question number 1, name the structure indicated by the arrows. So this is a section of mesentery, one of the loose areolar connective tissues. So these thick fuzzy bundles are collagen fibers, and these are the nuclei of fibroblasts mainly. And the arrow points into these well-defined dark fibers. These well-defined dark thin fibers are elastic fibers, which provide elasticity for this tissue. Question number 2, name the cell type outlined in black. This is a section of adipose tissue, one of the specialized connective tissues, and adipose tissue is composed of adipocytes. Adipocytes are large cells, and their cytoplasm is very thin, which gives the tissue a chicken wire-like appearance. And the nucleus is also found peripheral, it's pushed to the periphery. So the cells outlined are adipocytes. Question number 3, what is this a section of? Be specific. So in this section, there are few cells, but there are more fibers. And when you see the arrangement of fibers, the fibers are arranged in all directions. So it's a dense, irregular connective tissue. So when we come to today's lab session, the learning objectives are, by the end of this lab session, you will be able to describe the three types of cartilage, the hyaline, fibro, and elastic cartilage, and we'll see their structural difference and their function. And in regard to bone, we'll see the two types of bone, compact and sponge bone, and we'll see their structural difference. And we'll also cover the three types of bone cells, osteoblasts, osteoclasts, and osteocytes. And we'll also cover the two types of bone formation, the intramembranous and endochondral bone formation. So let's start with cartilage. So when you see the general organization of cartilage, cartilage is externally covered with perichondrium. Not all cartilages are covered with perichondrium. There are some exceptions. We'll see it later. But most of them externally, they are covered with perichondrium. And perichondrium has two layers, fibrous layer and chondrogenic layer. The fibrous layer is a dense, irregular connective tissue. And the chondrogenic layer, it consists of chondroblasts. So the fibrous layer, it mainly consists of type 1 collagen fibers. And there are fibroblasts that produce these collagen fibers. But in the chondrogenic layer, we have chondroblasts. These chondroblasts are derived from these fibroblasts. And the chondroblasts, as you see it in this picture, they undergo cell division and they give rise to new chondroblasts. And these chondroblasts, they produce matrix. And gradually they will be surrounded by the matrix and they will migrate into the main cartilage matrix and they become chondrocytes. But the production of the matrix and plus the cell division, it results in a growth. And the growth that happens at the chondrogenic layer of perichondrium is appositional growth. So the cell division plus the production of new matrix, these two processes result in appositional growth, and appositional growth happens at the chondrogenic layer of perichondrium. And when you see the main cartilage matrix, so there are chondrocytes which are found in lacunae. Chondrocytes are found in a space or in a depression which is known as lacunae. And these chondrocytes also undergo cell division, and they give rise to new chondrocytes. And they also produce matrix. And these two processes also result in other type of growth, which is known as interstitial growth. So interstitial growth happens at the cartilage matrix, but appositional growth happens at the chondrogenic layer of perichondrium. So when these chondrocytes undergo cell division, when they give rise to new cartilage cells or chondrocytes, the matrix is relatively rigid. It's not as rigid as bone. When we see bone matrix, it's very rigid. So even it doesn't allow the osteocytes to undergo cell division. But cartilage matrix is relatively flexible, but it doesn't allow the newly formed chondrocytes to migrate or to move. So most of the time, the newly formed chondrocytes, they will remain together and they will form isogenous group or cell nest. So due to the rigidity of the matrix, the newly formed chondrocytes, they cannot move, they cannot migrate, so they will stay together and they will form isogenous group or cell nest. And the matrix has two parts. The part that runs in between the isogenous group or the chondrocytes is known as interterritorial matrix. The part that surrounds the isogenous group or the single chondrocyte is known as territorial matrix. So as a summary, cartilage, most of the cartilage with some exceptions externally, they are covered with perichondrium, and perichondrium has two layers. Outer fibrous layer, which is mainly composed of type 1 collagen fiber, so it appears eosinophilic in HND staining because it's collagen fiber. The second layer of the perichondrium is the chondrogenic layer. It mainly consists of chondroblasts, which are derived from fibroblasts. And these chondroblasts, they undergo cell division and they give rise to new chondroblasts, and at the same time they produce matrix. And these two processes result in appositional growth. So this area indicates a growing cartilage or its appositional growth. And when we come to the cartilage matrix, in the cartilage matrix we'll have isogenous group or cell nests, and due to the rigidity of the matrix, the newly formed chondrocytes, they cannot migrate, they cannot move freely, so they will remain together and they will form isogenous group or cell nest. And the matrix has two parts, interterritorial matrix and territorial matrix. Interterritorial matrix, when we see it in staining, it appears lighter. But territorial matrix, it appears darker. And due to the amount of proteoglycans, we'll see it later, cartilage matrix mainly consists of proteoglycans. There are more proteoglycans in the territorial matrix, that's why it appears darker, but there are less proteoglycans in the interterritorial matrix. That's why it appears lighter in staining. So when we see cartilage matrix, generally cartilage matrix consists of proteoglycans and type 2 collagen fibers. It may vary depending on the type of cartilage, but generally it consists of proteoglycans and type 2 collagen fibers. So proteoglycans are proteins which are highly glycosylated. So they contain a core protein and sulfated glycosaminoglycan chains. These sulfated GAGs or glycosaminoglycans, they are negatively charged, so they attract hematoxylin. That's why cartilage matrix is basophilic. But when you see the type 2 collagen fibers, they are eosinophilic, but they are dominated by the proteoglycans. That's why cartilage matrix appears basophilic. But if it's an elastic cartilage, in addition to proteoglycans and type 2 collagen fibers, we will have elastic fiber. And if it's a fibrocartilage, in addition to proteoglycans and type 2 collagen fibers, we will have type 1 collagen fibers. So the matrix may vary, but generally it consists of proteoglycan and type 2 collagen fibers. But proteoglycans, they dominate type 2 collagen fibers. That's why cartilage matrix appears basophilic. It appears blue or purple in H&E staining. And it's due to the sulfated glycosaminoglycans. They are negatively charged. They attract hematoxylin, so they appear basophilic. Now let us see the three types of cartilage. So let's start with hyaline cartilage. So when you see some examples of hyaline cartilage in our body, we have tracheal cartilage, bronchial cartilage, some nasal cartilage, articular cartilage. These are some examples of hyaline cartilage. And the main function of hyaline cartilage is it provides support. For instance, the hyaline cartilage in the wall of the respiratory tract, it keeps our respiratory tract open, so it gives support. The other function of hyaline cartilage is it acts as a shock absorber. In some areas, some of the hyaline cartilage, they act as shock absorbers. It also reduces friction. And articular cartilage, it lubricates synovial joints. So these are some of the functions of hyaline cartilage. So when you see the matrix of hyaline cartilage, it's homogeneous. In a fresh section, it has a glassy appearance. That's why it's known as hyaline cartilage. It has a glassy appearance. And the chondrocytes are found in groups. And with the exception of articular cartilage, all hyaline cartilages are externally surrounded by perichondrium. But articular cartilage, it lacks perichondrium. So now let us see some slides from our virtual slide box. So first let us see trachea. So this is the longitudinal section and this is the transverse section of trachea. So let's zoom in and see the transverse section of trachea. So as you see it here, externally, it's surrounded by perichondrium. So this is the perichondrium. And let's zoom in more and see the two layers of the perichondrium. The part that appears eosinophilic is the outer fibrous layer. And this one is the chondrogenic layer. So the chondrogenic layer, as you see, it consists of chondroblasts. And these chondroblasts are derived from fibroblasts. And when we see the matrix, so as you see, it's homogeneous. And the chondrocytes are found in groups. So these are the chondrocytes, and they form isogenous group or cell nest. And the matrix has two parts. This is the interterritorial matrix, and the part that surrounds the lacunae, or the part that surrounds the chondrocyte, is the territorial matrix. And the territorial matrix, it appears darker, but the interterritorial matrix appears lighter, and it's due to the amount of proteoglycans. There are more proteoglycans in the territorial matrix. That's why it appears darker. So tracheal cartilage is surrounded by perichondrium. The other example of hyaline cartilage is articular cartilage. As I have told you, it's a special type of hyaline cartilage. And what makes it special is it lacks perichondrium. So this is a section of long bone, and this is the articulating surface of a long bone. This articulating surface is covered with articular cartilage. And when we zoom in and see the articular cartilage, so as you see it here, there is no perichondrium. So it lacks perichondrium. So as you know, cartilage is avascular. It doesn't have blood supply. So normally cartilage gets nutrition from the perichondrium by diffusion. But in this case, there is no perichondrium. So articular cartilage, it will get nutrition from the synovial fluid. It's found inside the joint cavity. So it will get its nutrition from the synovial fluid. But all the other features are the same. We have interterritorial and territorial matrix. We have isogenous group or cell nests. Now let us see elastic cartilage. So elastic cartilage is more or less similar with hyaline cartilage. The major difference is elastic cartilage, it consists of elastic fibers. So the matrix is not homogeneous. Due to the presence of these thin branching networks of fibers, it's not homogeneous. But the chondrocytes are found in groups, they form a cell nest, and elastic cartilage is externally surrounded by perichondrium. And the main function of elastic cartilage is flexibility. Due to the presence of these elastic fibers, it's highly flexible. So when you see some examples of elastic cartilage, the external ear or the pinna, it's highly flexible and is composed of elastic cartilage. Epiglottis, one of the cartilages in larynx, it's another example of elastic cartilage. So first let us see a section of epiglottis. So epiglottis is like a flap. When we swallow, it bends down and closes the respiratory tract. So it prevents the entrance of food from the respiratory tract. So as you see it here, elastic cartilage is also covered with perichondrium. So this pink band is the fibrous layer. And under the fibrous layer, this is the chondrogenic layer. And when we zoom in and see the main cartilage matrix, we have the chondrocytes. But as you see, the matrix is not homogeneous. These dark branching line-like structures are elastic fibers. But on this side also there is a perichondrium. So it's externally covered with perichondrium. The chondrocytes are found in groups, but the main difference with hyaline cartilage is the matrix is not homogeneous due to the presence of elastic fibers. The other example of elastic cartilage is the external ear or pinna. So this is the section of external ear or pinna, and this is stained with mesenteric and aldehyde fusion. Mesenteric, it mainly stains connective tissues, and the aldehyde fusion stains the elastic fibers. So let's zoom in and see. So this is the perichondrium. So mesenteric, it stains connective tissues blue. So this is the outer fibrous layer of the perichondrium, and this one is the chondrogenic layer of the perichondrium. Let's zoom in and see the chondroblasts. So these are the chondroblasts, and this is the collagen fiber. And these thin branching or networks of fibers are elastic fibers. So the elastic fiber is stained with the aldehyde fusion. And on this side also we have the perichondria. Now let us see the third type of cartilage, which is fibrocartilage. So fibrocartilage, like hyaline cartilage, the matrix is homogeneous. And in addition to proteoglycans and type 2 collagen fibers, fibrocartilage contains type 1 collagen fibers. So that's why the matrix appears eosinophilic in this section. And in fibrocartilage, the chondrocytes are arranged in a row, or they are arranged linearly. In elastic and hyaline cartilage, the chondrocytes are found in groups. They form isogenous group or cell nest. But in fibrocartilage, the chondrocytes are arranged in a row. But in addition to chondrocytes, we can see the nuclei of fibroblasts. So these fibroblasts, they produce type 1 and type 2 collagen fibers. And when you see some examples of fibrocartilage, we have the outer part of intervertebral disc, and there are also articular disc. Inside some joint cavities, there are pieces of fibrocartilage. These pieces of fibrocartilage, they act as shock absorbers, and they are known as articular disc. And generally, the main function of fibrocartilage is for shock absorption. So when we see an example of fibrocartilage, the outer part of intervertebral disc is fibrocartilage. One of the unique features of fibrocartilage is it lacks perichondria. Fibrocartilage, it doesn't have perichondria. So when you see a section of fibrocartilage, this is the outer part of the intervertebral disc, or the annulus fibrosus of intervertebral disc. So when you see the matrix, we'll see basophilic and eosinophilic areas. Where the proteoglycan dominates, the matrix becomes basophilic, and where the collagen fibers dominate, the matrix becomes eosinophilic. And these are chondrocytes. And as you see, chondrocytes are arranged in a row or in a column. And the matrix near the chondrocytes is basophilic, and it's due to the amount of proteoglycans. So as you see it here, the chondrocytes are arranged in a column, or they are arranged linearly. And this is where the collagen fibers dominate, and this is where the proteoglycans dominate. And as you see it here, it's not covered with perichondrium. It lacks perichondria. So fibrocartilage lacks perichondria. Now let's see the other type of specialized connective tissue, which is bone. So histologically, there are two types of bone. We have compact bone and we have sponge bone. So spongy, as the name indicates, it looks like a sponge. That's why it's known as spongy or cancellous bone. So sponge bone, it consists of thin columns of bones, which are separated by unequal spaces. These thin columns of bones are known as trabeculae or bone spicules. And the space in between these bone spicules is filled with bone marrow. And when you see compact bone has a very organized appearance. It's composed of osteons. So osteons are cylindrical structures, and a single osteon consists of central canal or Haversian canal and a surrounding lamellae. So the lamellae that surrounds the central canal or the Haversian canal is known as concentric lamellae. And in between the concentric lamellae, there are interstitial lamellae. These are leftovers from bone remodeling. And we have also circumferential lamellae. So as their name indicates, these circumferential lamellae, they run throughout the circumference of the bone. They lie immediately under the periosteum. And like cartilage, bone is also covered with periosteum externally. And periosteum has two layers, outer fibrous layer and inner osteogenic layer. It's homologous with perichondrium. Perichondrium has also two layers, outer fibrous and inner chondrogenic. And in addition to periosteum, bone internally it's lined with endosteum. So the bony spicules of sponge bone and the canals, central canal or the main medullary canal, they are lined with endosteum. So externally it's lined with periosteum and internally it's lined with endosteum. And endosteum, it consists of osteoprogenitor cells and osteoblasts. And connecting adjacent central canals or running perpendicular to these central canals, we have a Volkmann's canal. And these central canals are pathways for neurovascular structures. So first let us see compact bone. Let's see some sections of compact bone. So this is a section of compact bone. It's a ground section. It's not stained. It's a very thin layer of bone, which is filled with India ink. And these are osteons. So let's zoom in and see one of the osteons. So let's see this one. So this is the central canal through which the neurovascular structures run. And surrounding this central canal, this is the concentric lamellae. So this is the concentric lamellae. And in between the concentric lamellae, there are small spaces which are known as lacunae. And these lacunae are occupied by osteocytes. A single osteocyte is found in each lacunae. Bone matrix is very hard, so it doesn't allow the osteocytes to undergo cell division. The lacunae, it doesn't have a space for another osteocyte. So due to the rigidity of the matrix, the osteocytes, they do not undergo cell division. So a single osteocyte is found in each lacunae. But when we see cartilage, if you remember, the matrix, it allows the chondrocytes to divide, but it doesn't allow them to migrate. So they will remain together and they will form isogenious group or cell nest. But it's not that much hard to restrict them from dividing. But bone matrix is very rigid. It doesn't allow the osteocytes to undergo cell division. And running from each lacunae, there is a very thin canal, which is known as canaliculi. This canaliculi, it allows communication between adjacent osteocytes. Osteocytes, we'll see it shortly. They have several cytoplasmic extensions. They have several cellular processes. So these processes, they run through these canaliculi and they communicate with adjacent osteocytes by gap junction. So one of the function of osteocytes is trafficking of nutrients and wastes. So adjacent osteocytes, they communicate through these canaliculi, these very thin hair-like canals, which are known as canaliculi. But in between these concentric lamellae, these are interstitial lamellae. Interstitial lamellae are leftovers from bone remodeling. We have also another slide of compact bone with a different staining, but the structures are the same. This is with shimmer staining. So this is the central canal, and this is the concentric lamellae, and these are the lacunae occupied by a single osteocyte, and these thin canals are known as canaliculi. And canaliculi allows communication between adjacent osteocytes. And these are interstitial lamellae. Now let us see sponge bone or cancellous bone. So first let us see section MH47. It's a section of spinal cord, but our main concern is not the spinal cord. We'll see the spinal cord in a later lab. But covering and protecting the spinal cord, we have the vertebral column. So let's see this vertebrae. So as you see, it consists of these thin columns of bones, or these are bony spicules or trabeculae. And in between the trabeculae, this highly cellular structure is bone marrow. And these bony spicules, as you see it here, let me zoom in more. It's lined with endosteum. And endosteum is composed of osteoprogenitor cells and osteoblasts. We'll see shortly how osteoblasts look like and their function. But this is the sponge bone. We have also another slide which contains both compact and sponge bone. Let's see this one. So the outer part is compact, and the inner part is the sponge part. And when we see bone in H&E staining, first it needs to be decalcified. So the inorganic component will be removed. So the main organic component of bone is type 1 collagen fiber. So in H&E staining, bone appears eosinophilic. If you remember, cartilage appears basophilic, it appears blue, because its main content is proteoglycan, which contains sulfated GAGs, and sulfated GAGs are negatively charged, so they attract hematoxylin. But when we see bone, the main organic component of bone is type 1 collagen fiber, and as you know, collagen fibers are eosinophilic. So bone appears pink in H&E staining. So this is the spongy part of this section. So these are the bony spicules, which are lined with endosteum. It's not shown here, but these spaces will be filled with bone marrow. Now let us see the three types of bone cells. So first let us see osteoblasts and osteocytes. So osteoblasts are derived from the osteoprogenitor cells. And the main function of osteoblasts is to produce bone matrix. So osteoblasts, they produce bone matrix. And this newly formed bone is known as osteoid or pre-bone. It's not calcified. It has only the organic components, so it appears lighter in staining. So as you see it in this diagram, the lighter part is the newly formed bone, which is produced by osteoblasts. When it becomes calcified, it will be darker. So when the osteoblasts are inactive, they become flat in shape. So when they are active, they are locuboidal. Gradually, these osteoblasts, they will be surrounded by the newly formed matrix, and they become osteocytes. And a single osteocyte is found in each lacunae. So let's see these osteoblasts in a section. So you can see it here. This is a bony specule, or it's a sponge bone, and it's lined with endosteum. And endosteum, it consists of osteoprogenitor cells and osteoblasts. So these are active osteoblasts. And these active osteoblasts, they produce osteoid or prebone. This osteoid or prebone, as you see it appears lighter in staining. When it becomes mineralized, it will be darker in staining. So when they are inactive, they are flat, but when they are active, they become low cuboidal in shape. So the main function of osteoblasts is production of osteoid or prebone. So they are involved in bone remodeling. So gradually, when they are surrounded by the matrix, they become osteocytes. So osteocytes are found in each lacunae. And the main function of osteocytes is to maintain the matrix. They maintain the health of the matrix. And as I have mentioned earlier, osteocytes, they communicate through the canaliculi with adjacent osteocytes by gap junction. So they traffic nutrients and wastes between these osteocytes. The other main function of osteocytes is they act as mechanoreceptors. They sense mechanical stress, and they signal this into osteoblasts or osteoclasts. Depending on the need, they stimulate the osteoblasts to produce new bone matrix, and they stimulate the osteoclasts to resorb bone tissue. So osteocytes, they communicate both osteoblasts and osteoclasts, and they act as mechanoreceptors. And they also maintain the matrix, the health of the matrix. And they also transport nutrients and wastes. The other type of bone cell is osteoclast. Osteoclasts are derived from monocytes. They are formed by the fusion of several monocytes. So osteoclasts are multinucleated cells. And osteoclasts are found in a space known as Hauschup's lacunae or resorption bay. So let's see osteoclasts in a slide, a section. So this is a section of a long bone. And in this area of the long bone, or below the epiphyseal plate, there is a higher bone remodeling in this area. So osteoclasts are mainly found in this area. So here there is one osteoclast. Here there is another osteoclast. So as you see, they are multinucleated, and they are found in Hauschup's lacunae or resorption bay. And their main function is for resorption of bone tissue. So osteoblasts and osteoclasts, they work hand in hand. Bone is very dynamic. It might look like a static one, but it's very dynamic. So osteoblasts lay down new bone tissue, and old or unwanted bone tissue is resorbed by osteoclasts. Now let us see bone formation or ossification. There are two types of bone formation, intramembranous and endochondral bone formation. So in intramembranous bone formation, the embryonic connective tissue or the mesenchyme, it consists of stem cells as you know it. So this embryonic connective tissue or these mesenchymal cells, they will aggregate and they will differentiate into osteoprogenitor cells. And these osteoprogenitor cells, they will give rise to osteoblasts, or they will differentiate into osteoblasts. And osteoblasts, they produce and lay down new bone tissue. And gradually, these osteoblasts, they will be surrounded by the matrix and they become osteocytes. So this type of bone formation is intramembranous bone formation. So the upper part of our skull, or the calvaria, the maxillae, mandible, the main part of mandible, clavicle, they are formed by intramembranous ossification. But when we see endochondral ossification, bone is formed from cartilage. So first we will have a cartilage model, and that cartilage model will be changed into a bone. So most of the bones in our body are formed by endochondral ossification. The long bones in the limbs, the vertebrae, the pelvis, the base of our skull. Most of the bones are formed by endochondral ossification. So now let us see some slides from our slide box. So first, let us see intramembranous ossification. So this is the section of an embryonic face. So here there is a mesenchyme, an embryonic connective tissue. So some of the mesenchymal cells, the stem cells, they aggregate and differentiate into osteoprogenitor cells. And osteoprogenitor cells, they differentiate into osteoblasts. So these are active osteoblasts, which are locuboidal. And these active osteoblasts, they produce matrix. And gradually, some of these osteoblasts, they will be surrounded by the matrix and they become osteocytes. And as you know, osteocytes, they will maintain the matrix. Or you can see it here. These are osteoblasts, active osteoblasts, which lay down new bone tissue. And these are osteocytes. So such type of bone formation is intramembranous. And now let us see the endochondral. So let's see the epiphyseal plate. After the long bone is formed between diaphysis and epiphysis, we have epiphyseal plate, a piece of hyaline cartilage. This epiphyseal plate allows longitudinal growth of bones. And the epiphyseal plate has 5 zones. Now let us see these 5 zones in a section. So this is a section of long bone. So this is the epiphysis and this is the diaphysis. So developmentally, first we have a cartilage model, and the first site of ossification is the diaphysis. So the primary ossification center was at the diaphysis, and the secondary ossification center was at the epiphysis. So between diaphysis and epiphysis, we have this epiphyseal plate. And this epiphyseal plate, it allows longitudinal growth of bones. And the epiphyseal plate has 5 zones. So now let us see these 5 zones of the epiphyseal plate. So the first zone, which lies adjacent to the epiphysis, is zone of reserve or zone of resting cartilage. So this is zone of resting cartilage. The second zone is zone of proliferation. So the chondrocytes, they undergo cell division and they will be arranged in columns. They look like stacks of coins. So this is zone of proliferation. The third zone is known as zone of hypertrophy. Now the chondrocytes, they don't undergo cell division. They become large in size. So that's why it's known as zone of hypertrophy. So this is zone of hypertrophy. The fourth zone is known as zone of calcification. The cartilage matrix, it becomes calcified. So when it becomes calcified, chondrocytes, they will not get nutrition. Normally, chondrocytes get nutrition by simple diffusion. But when it's calcified, it's hard to get nutrition. So the chondrocytes, they undergo apoptosis, or the chondrocytes will die. The fifth zone is zone of resorption or zone of ossification. Now, blood brings osteoblasts and osteoclasts to this area. So osteoblasts, they will begin to produce bone tissue, and osteoclasts, they will resorb unwanted part. So the fifth zone is zone of ossification. So here we can see these low cuboidal cells, which are osteoblasts, and you can also see it here. These are osteoclasts. We have also another slide of this epiphyseal plate. Let's zoom in and see. So this is resting zone. Then this is zone of proliferation. As you see, the chondrocytes are arranged in columns, or they look like stacks of coins. Then the third zone is zone of hypertrophy. In this zone, there is no cell division, but they become larger in size. Then there is zone of calcification. The matrix becomes calcified. So when the matrix is calcified, the chondrocytes will have difficulty to get nutrition, so they will undergo apoptosis or cell death. Then the last zone is zone of ossification or resorption. So blood, it brings osteoblasts and osteoclasts. And as you see here, these are osteoblasts, and the osteoblasts, they began to produce bone tissue. They will lay down a bone tissue. And the osteoclasts, they will resorb the bone tissue. So this epiphyseal plate, it allows longitudinal growth of bones, and it lies between the epiphysis and the diaphysis. And the primary ossification center was found at the diaphysis, and the secondary ossification center was found at the epiphysis. So you can use this slide to summarize the epiphyseal plate, the zones of the epiphyseal plate. So this is the zone of resting cartilage, zone of proliferation. Here we have zone of hypertrophy. Here there is zone of calcification, and this is zone of ossification or resorption. So that's all about today's lab session. We have covered all these sliders. So study these sliders and practice identifying structures. And good luck on your assignment, and I will see you next lab.